Our next speaker, moving along with our Italian program, is Tiziana Brevini. And Tiziana is now teaching anatomy and neuroanatomy, right, at the University of Milano. But she has a very interesting history. She spent a number of years in and out of Melbourne, Australia, working with Alan Trounston and some other pioneers. She is a pharmacist by training. She took a pharmacy degree in Milan, and then she went to Cambridge to do some of her work, Cambridge, England, not Cambridge, Massachusetts. And then she has been in Melbourne and in Adelaide. She worked with David Armstrong and as I said, with Alan and still collaborates. And she has a project going on in Florida that has to do with, I don't remember. Yes, that's right, pancreatic stem cells. And so she's in and out of Florida now a little bit. And she's going to talk to us today on because our theme is circadian rhythms and parthenogenesis. And so, Tiziana.
Thank you for coming. Thank you very much. Thank you for your kind introduction, Anne, and yeah, thank you for this opportunity to share with you results obtained in our laboratory. We had a special interest in trying and elucidate the specific contribution of the egg to embryo developmental competence with looking at the different sorting of molecules that may involve in rendering the oocytes able to support development. To do this, we used mostly large animal models. We have been using large animal models in years, and parthenogenetic activation was one of the protocols that we were using indeed in trying to understand the role of the oocytes in development. But in February 2004, the Italian Parliament promulgated a law, which is Law 42004, which set out the rules for assisted reproduction technology in our country. And this law had a number of restrictions, not going into, but basically, to our concern, is the part where the law banned the creation of more than 3 embryos at once per cycle per patient. This, of course, led to a very strange situation where, in most cases, patients would produce more than 3 oocytes, and there was a number of supernumerary oocytes that were not used. And therefore, at that stage, women were offered either to cryopreserve the supernumerary oocytes, or they could donate them to research. So in a way, Law 4204 gave scientists, and our lab as well, the unprecedented access to a large number of supernumerary oocytes. Of course, we only used oocytes that were not fertilized, neither frozen, and that were donated by the patients, and no extra treatment was involved. And I think this is a very important aspect to stress. So this is how we found ourselves in a very privileged position, because we had a well-established protocol of parthenogenetic activation going in the lab, and we suddenly had a large number of oocytes. So this is the first part of the experiments. Then we had more oocytes coming, and then the law was slightly modified, and so the number of oocytes that became available decreased progressively in the years. So this first attempt gave us access to 104 oocytes altogether, human oocytes, that were subjected to parthenogenetic activation, and 61.5% of them cleaved, and 12.8 developed to the blastocyst stage. And this is our first human parthenogenetic blastocyst that was derived at the moment. And we, of course, published the results, and we compared the results in terms of morphology and also all sorts of parameters, comparing parthenogenetic activated blastocysts versus ICSI obtained embryos. Of course, this was very exciting because it gave us the opportunity to acquire fundamental knowledge on human blastocysts, but also gave us the opportunity to generate stem cells from these blastocysts. And the idea behind that was not only to carry out experiments of fundamental research on a material that was, of course, more abundant because it was obtained from cell line, but also to create an alternative, ethically acceptable stem cell line. I remind you, at the time, IPS were not around yet, nor chemical reprogramming. So most of the regenerative medicine related approach was based on embryonic stem cells, which of course implied a number of ethical concerns and objections. So basically, we subjected the parthenogenetic embryos to the isolation of the ICM. The ICM were plated on inactivated feeder layer, exactly as we would do with embryonic stem cells. And with that series of experiments, we were able to generate 3 human parthenogenetic stem cell lines, which we arbitrarily defined HP1, 2, and 3. Well, we knew that this was something that was very much needed because there was a great expectancy in terms of finding an alternative way to have access to stem cells, to cells that would offer plasticity and differentiation potential. But in a way, we were not ready to the surge of interest that we received when we firstly presented our results at the ESHRE conference in Prague. That was 2006. And this interest came both from the specialized press, but also from the very general one. So of course, it appeared to be something that was very felt and very needed by ordinary people as well as by specialized people and scientists. What we were completely unprepared was this. We were invited by the Pontifical Academy for Life, and this is Benedict XVI, and they wanted us to explain exactly what was behind this approach and why these cells would be ethically acceptable, which was a very particular experience, I have to say, but very, very interesting as well. Anyway, so now let's go back to the cells. This is just the history and nice rememberings. So what are these cell lines like? So since then we were able to derive several cell lines. 2 of these lines have been in culture for a long time. They underwent 71 passages and 407 doublings. They showed stable morphology, good proliferation ability. They could undergo repeated freeze-thawing cycles with no problem whatsoever. So we were very pleased by these cell lines. I would like to spend a little bit telling you about the pluripotency of these cell lines. So we could show that they express the main pluripotency markers such as OC4, SF4, NANOG, and so on, and no differentiation markers when they are kept in culture in pluripotency related medium. They have telomerase activity as we heard this morning of slightly earlier stages. They have normal telomerase activity, and they switch off telomerase activity when they are addressed and harnessed towards differentiation, which is what we expect to happen. If we then allow them to differentiate spontaneously, differentiation by capturing them in a hanging drop system, we get very beautiful embryonic bodies that pick up the expression of the three germ layer markers. We just selected these three, but we could have used any other. And you can see this both in terms of PCR and immunocytochemistry for endoderm, ectoderm, and mesoderm, showing the plasticity of these cells. On the other hand, if we harness their differentiation using a specific and distinctive differentiation protocol, we were able to get all the three cell types of the hematopoietic lines. This was done in collaboration with the cell factory of Ospedale Policlinico in Milan. And similarly, we could show positivity and induction of the main neural progenitors that were with cells that would express positive, would be positive for nesting, for MAP2 and CNPAs, to all the different neuronal population. So this was quite good, at least in our understanding. And to conclude this very early part, we are convinced that oocyte parthenoids can be used as an alternative source for establishing and generating pluripotent cell lines. As we have seen, these cell lines have great differentiation ability. They give rise to cell types, different cell types in the body, and they generate the three germ layers. So all these in general display, allow us to say that they display many characteristics that are common to biparental cell lines. However, we also observe that these cells possess distinctive properties, which can be of interest of course. For instance, we noticed that we found on a regular basis multiple centroids. So basically we would detect, and you can see these little green spots. These are centroid positive spots, and this is electron microscopy, and we could detect a variable number of centrioles in our cells, varying in general from 1 to 5. So of course, this is, for instance, the kind of results we get with our human parthenogenetic cell lines, and the differences in terms of number of centrioles occurring in cell population. So the next thought, just considering this, we started to hypothesize this might be related to the age of patients. Patients were comprised between 32 and 39 years of age. So we went back to the same protocol, and we subjected we used the same protocol, but this time we used pig as a source. So this is the result we got from pig parthenogenetic cells. And as you can see, the number of centroids occurring in cells, again, is variable between 1 and 2 centrioles per cell. So this obviously indicated that something else was there, which was not related strictly to the age of the patient that were involved in the experiments. So of course, the next step was maybe we are doing something wrong, culture procedure, cultural artifacts, which may very often happen, and technical artifacts. So we decided that one way to answer this point was to take another approach. And together with our colleague Sergio Leda from the University of Sassari, they have a very good access to sheep because Sardinia is an island and they have a lot of sheep and many other beautiful things that you find there. So we basically, our colleagues went to the slaughterhouse, they collected oocytes that were matured in vitro and activated exactly with the same protocol that we have been using for the human and the porcine. And then the presumptive parthenogenetic zygotes were cultured for a few days and then transferred to the uterus of synchronized sheep recipients. And then we let gestation continue for a number of days. And the number of days we allowed was the 27 days, because this is considered to be the limit of development of parthenogenetic sheep. So we knew in vivo. So we knew that if we were keeping gestation longer, we would probably have degeneration and death. So we wanted to have nice fetuses growing in the recipient. And then just after that, cells were isolated from the fetuses and looked for, evaluated for central number and of course another series of parameters. These are very nice images that Sergio took. This is the fetus in the uterus developing very nicely. This is very impressive for me because this is a parthenogenetic fetus growing very nicely. They had heart beating. They had a number of very nice aspects, but they were parthenogenic, 100%. And we then went back to the centrioles, to these cells, and we found again a variable number of centrioles in these cells obtained from parthenogenetic fetuses. Of course, we can think of different explanations for this. For sure, we could see that altered expression levels of genes, the main genes that are related to central duplication and central organization, were abnormally regulated. In parthenogenetic here are blue bars and red are the parental cells. And you can see that most of these genes like CNPE, polo-like kinase, BUB1, MET2, that are all the genes that are directly involved in the control of central duplication are dysregulated in parthenogenetic stem cells. So our theory here, but of course it's a theory, is that there is a possibility that the absence of the sperm, which is not bringing the centrosome with fertilization, dealing with Parthenon, is probably creating a situation where we do not have the preparation and setting of a normal mitotic spindle. As we know, during oogenesis, centrioles degenerate during maturation. And this is true for all higher species animals, with the notable exception of the mouse. So this doesn't happen in the mouse. So basically, what the oocyte is left at the end of the maturation is a degenerated mass of centrosome, centrosomal material, which is not correctly organized as a centriole. And usually fertilization brings this centriole and rearrange the mitotic spindle, the correct mitotic spindle in the cycle, giving the template for the correct organization of the first mitotic spindle following this. So we think that a possible explanation for the wrong number of centrioles is exactly what may very likely be this. Also because the number of centrioles is considered to be to have an inhibitory effect on the formation of new centrioles. So if you have the right number of centrioles, you don't form more centrioles. But you keep producing centrioles, if you don't have the right template there, at least according to a number of reports. So in general, in lower species, the number of centriole and the formation of a correct spindle is considered to be the main strict control, checkpoint control to suppress, if needed, parthenogenesis, which would lead to accumulation of genetic anomalies and diseases and so on. In higher mammal, probably this control is much less stringent because we have genomic imprinting, which makes its job. That cleans the situation and prevents this to happen. And so probably the control and the patrolling is a bit more slack. And having genomic imprinting that as a main mechanism that ensure the parental fertilization, there is a possibility that the central duplication becomes something which is likely to happen. So what is the consequence of centrosome amplification? Well, basically, this is known. We didn't discover anything. The consequence of this is that multipolar division are likely to happen. And so there is a high possibility of aneuploidy in the project because the material will be pooled with irregular strength towards different direction, which is something which is in contrast ensured by a normal mitotic spindle. And indeed, when we look at chromosome instability, in all the cell lines I told you about, so human, pig, and sheep, we see a number of different setups in terms of number of chromosomes, which is, in our opinion, a bit more abundant than the one expected in general in embryos. So, however, this is not something which is confined to our cell lines, because this has been described, a high incidence of misshape and aneuploidy has been described in parthenos derived from many other groups in different species. All displaying abnormal chromosomal complements, and that have been shown very early on from the very first cell cycles. So each of these reports are somehow connecting and linking the recurring of abnormalities in parthenoids to the absence of paternally supplied centrosome. So somehow the sperm, beside all the very important aspects that we have heard in previous speakers, is somehow giving the correct symmetry for the organization of the first mitotic spindle in the zygote. So this is to conclude this part. Sorry, I made a mess. Yes. So we think that multiple centrosomes occur in parthenogenetic cells, and we suggest that the absence of paternally supplied centrosome prevent the formation of a correct spindle. This aspect, which has been considered and suggested as a strict checkpoint in lower species, is probably more likely to happen in higher species where genomic imprinting has the main role in this relation. Okay, but the next question is, so how do we get this beautiful looking evidence? How is it possible that we get this beautiful cells, fantastic differentiation, stable cell lines, but still we do have this problem with mitotic event. Well, we were very much intrigued by this, and we continued our experiments. And one of the aspects that was really interesting to us was that parthenogenetic cells, instead of being blocked along the cell cycles, as one would expect, looking at the level of the mitotic spindle checkpoint proteins, for instance, which were very abnormally expressed, would sort of bypass this and continue proliferating. And we found that one interesting aspect was the persistence of intercellular bridges. So here you have some very nice images where you can appreciate that these cells do organize intercellular bridges. And we were again very surprised by that. And we started to ask, how is this possible? And we thought that one possibility behind that could be found in the idea that if cells would divide material in a non-even way, but due to this abnormality would share material in non-uniform way, one way to solve the problem would be to interact one another, make a team and have an even and stable system that would work in a stable way. And so we started to look at the expression of genes and factors that are involved both in stabilizing intercellular canals or bridges, such as Aurora B and Edge-like 3, but also to molecules that are well known to shuttle, to be able to encourage shuttle of protein and other material through intercellular canals, such as translink, for instance. And you can see that, once again, parthenogenetic cell line blue bars do have a dysregulated, upregulated expression of all these molecules, somehow supporting the idea of this persistence intercellular bridges there. Well, are this canal working? The only way to answer this question was to use dextran. We selected dextran 10 kDa, which and we microinjected this in the cytoplasm of a single cell, and then we looked at what was happening to this tracker. And we could see that the tracker would extensively move along the canals to the nearby cells, indicating that these intercellular bridges are not only morphological, beautiful structures, but they are functional, and they allow cell-to-cell functional trafficking among the Symposium or cell population. So the next question was, are these intercellular canal really useful? What happens if we knock down, for instance, translink, which I told you is one of the molecules, a key molecule for allowing trafficking along the intercellular canals. And we saw that when we knock down translink for 24 hours, we disrupt intercellular bridges irreversibly. You can see extensive vacuolization of cells, and very soon cell dies. And they stop proliferating. In contrast, if we use knockdown, if we knock down translink for a shorter period, such as 12 hours, we can see that there is some damage, but there is the effect is temporary. And if we leave a 7-day recovery after removing the translink siRNA, cells re-establish the canals and cell return to normal trafficking and normal proliferation, somehow suggesting that these canals are functional to keep this cell population active and thriving. So to conclude this part again, we suggest that intercellular bridges ensure cell-to-cell trafficking in parthenogenetic cell lines, and this allows cell lines to work in cooperation and to overcome the problem related to an uneven distribution of material in cells. And also we show that disruption of these bridges causes hospitalization and death, suggesting that, once again, that these canals play a key role in this respect. Well, another very strange thing, and we have no explanation whatsoever for this, was any significant hyperchromicity in the DNA of these cells. So if we look at some stretches of DNA, such as huge palindromic element, we could see that we needed a much higher energy to open up the double strand of parthenogenetic DNA versus biparental DNA. And again, we have no explanation for this. And of course, I'm looking forward to hear if you have suggestions of any sort in this respect. And the next step, this was done in collaboration with Nissim Ben-Benisti from the Silberman Institute in Israel. Nissim had very nice biparental human embryonic stem cell lines. And together we compare the expression pattern of two parthenogenetic cell lines, two of our cell lines, HP1 and HP3, with the expression pattern of his cells. And of course, this was a huge number of data, and I'm not for sure going into all this. We have no time, and it would be probably very boring. But the gene clustering analysis gave a number of indications, and I would like to draw your attention on a few ones. For instance, raw signaling, mechanosensing related molecules, and cell adhesion and cell pluripotency. Okay, well, we already know about cell pluripotency. We already saw a little bit of results related to this. I would like to tell you a bit about this raw signaling story. And we then focused on specific genes coming from the Affymetrix results. And we could see that for sure, basically here, green is parthenogenetic, red is biparental. As you can see, most of the genes that are activating the raw signaling pathway are upregulated, upper histogram, are upregulated in partners. In contrast, most of the genes that are acting as inhibitors are downregulated in the parthenogenetic cells versus the biparental, suggesting that somehow there is a boosting effect on the raw signaling pathway. So raw proteins are belonging to the RAS superfamily. They play a role in cell polarity, cell shape, cell adhesion. And they also have a direct effect on one of the main pathways that put together cell response and mechanosensing relating effect. So basically, how cells respond to mechanosensing effect, physical strength, pressure, and all sorts of mechanosensing effect that may derive from the environment. So this pathway, which is the Hippo pathway, responds in a very distinctive way to the raw protein families, and therefore putting in relation the two aspects. So the Hippo signaling is a bit complex, but I will just very briefly tell you about it. So the raw protein are acting on the Hippo signaling, which is a very complex pathway. Basically, the main actors here are this factor, LATS and MOB, and these two do induce the phosphorylation of the two main actors of the Hippo signaling, which are respectively YAP and TAS. So basically, what RAW does is affect the phosphorylation of these two molecules, and these two molecules will either phosphorylated or not induce the phosphorylation of YAP-TAS. The result of this, the final effect of this, to cut a short story, the long story short, is that if YAP-TAS is phosphorylated, cell enters a differentiation state, and YAP-TAS is compartmentalized to the cytoplasm. When YAPTAs are not phosphorylated, they may translocate to the nucleus. They usually translocate with other factors such as SMAD and TER, and this allows the activation of proliferation, and this is the configuration, the nuclear configuration of all this system, enhanced self-renewal, pluripotent, and so on. So basically, this configuration is specific and distinctive of self-renewing pluripotent cells, while cytoplasmic compartmentalization of YAPTAs, generally phosphorylated, will enhance differentiation of cells and prevent pluripotency. So here we have a situation where raw signaling, the raw signaling pathway is, as you can see, very much upregulated in parthenoids. Again, green bars. So we have a lot of raw, 4 or 5 more. Then I'm done. Okay. As you can see, all the molecules related to the hippo pathway are statistically comparable in parthenoids versus biparentals. So they are there. They can respond in both ways. However, if we look at, and in a similar way, LUTs, which is the main one, which is target for raw, is statistically comparable. However, if we look at its phosphorylated form, it's much lower in parthenogenetic versus biparental cell. Oops. Okay, so again, so basically what we see is that the trend will be to have a phosphorylated YAPTAs, but to inhibit, the phosphorylation of YAPTAs in a situation where we have a higher concentration of the YAPTAs gene. So the final result is that we have an increase in total YAP, but a non-significant change in the level of phosphorylation, suggesting that YAPTAs remain unphosphorylated and it may translocate to the nucleus. And when it translocates to the nucleus, this is in general what happens. So YAPTAs nuclear localization co-translocate SMAD2, and the co-translocation of YAPTAs and SMAD2 enhance the transcription of the pluripotency gene. And this is the other situation, which is typical of differentiating cells. So basically, we are dealing with a situation where the parthenoids have the ability to translocate YAPTAs to the nucleus, enhancing the pluripotent related pattern expression in cells. Right, and indeed, this is what we see. If we look at the expression of the main pluripotency gene, they are much higher in parthenogenetic cells than biparental cells. And as expected, we have a much decreased expression of DNA methyltransferases, which are the enzymes that methylate DNA. So basically, the DNA is maintained in an hypomethylated state, which is coherent with the expression of pluripotent genes. And consistent with that, the global DNA methylation is lower in parthenoids versus biparental cells. So if we look in a very general kind of table where we see the active and passive demethylation event in mammalian cells, we can see that basically the parthenoids favor and replicate the methylating waves that decrease the NMT activity and thus keep the system somehow returning into these phases, somehow discouraging the progression towards the removal of epigenetic asymmetry and from the gamete imprinted DNA. So again, to conclude this part, we just saw that YAP-TAZ is upregulated and remain rephosphorylated, therefore confined to the nuclear localization in parthenogenetic embryonic stem cells. And nuclear localization, as we have seen, boosts pluripotency. So this, of course, may account for parthenogenetic higher ability to form outgrowth, to keep pluripotency and proliferate for a long time, to form 3D spheroid colonies, and to show high level of high plasticity related genes. Since YAP mRNA is expressed in the oocyte and is maternally inherited, persistence of YAP can be a regulatory mechanism that parthenogenetic embryonic stem cells may display in relation to their strictly maternal origin. On the other hand, this property may be used in order to boost all those stimuli that may derive from mechanosensing related forces. So we are now using scaffolds and microbioreactors to replicate this response. And so basically to encourage the YAP-dependent effect, which, by the way, has also been shown in the mouse IVF and oocyte culture, where the addition of oocyte maturation and embryonic and embryo culture, where the addition of YAP activators have been shown to increase the efficiency of the process. Okay, with that, this is a very general conclusion. We have seen that these cells have high differentiation ability. They have many features that are common to biparental cells. However, they have distinctive properties such as the supernumerary centrioles and the lack of the paternal centrosome. Therefore, this leads to multipolar spindle and some chromosomal instability, a specific expression profile, and active pathway that are strictly related to their maternal origin. And these are all the funds that were that made this research possible in the years. And I would like just to thank all the people that were involved in this research. For sure, as already Anne mentioned, Alan Transon, who was encouraging with never-ending strength these experiments, Sergio Leda from the University of Sassari for the sheep experiments, all the people in my lab, Nisim Brevini for the collaboration of the Affymetrics, and Fulvio Gandolfi, who's also the PI of our lab, with whom I share many years of laboratory work and also personal life. Thank you for your attention.
Great. We have time for a couple of questions.
So, right. So this multiple centriole and multiple centrosome thing is interesting because your analysis of the parthenogenesis in the human actually, in some ways, leads us back to Ozil's first experiments with rabbit. He was able to use a pulsatile electrical activation of rabbit eggs to get mid-gestation fetuses that were normal, had a beating heart and pretty much everything in shape. And the sheep results show pretty normal fetus. So how do you get there when you can't regulate your centrosome? That's the question. And the reason I bring it up is because I think we all now are accepting the fact that this kind of genetic chaos, spindle abnormalities, etc., is the norm for the human embryo. Okay, and as the argument goes, maybe for the inner cell mass it corrects itself. We hope, but for the trophoblast, it obviously does not. And that supports it. So how do you tie that to getting a normal fetus
I do
out of a
agree.
genetically abnormal
I do,
cell?
I do agree 100% with you. My impression was that we were kind of harnessed in a situation where we were supposed to show that these cells had the right setup of chromosomes and had the right setup of centrioles, which was not the case. And but on the other hand, I don't want to be abrupt, but when you submit a paper and you know present this kind of evidence, where the response cannot be, I have what you just said, that, you know, for sure, there are possibilities that all this about genetic stability and all these aspects probably are not so restraining, that there are adjustments that are there. And for sure, I mean, looking at this sheep, these sheep fetuses were absolutely normal and probably just slightly smaller instead in terms of sizes. But, you know, it was not significantly smaller. We made a few of them, so we know that. On the other hand, you know, this is the rules and you have to play by the rules. So until we will be told that, okay, chromosomal instability is not a problem, the problem is there. I have to say that, of course, one way to answer the question would be to microinject the central. Okay. To microinject the central.
Right.
We never had the permission to do it in human. Even if we applied for the permission to just, together with Cesare Galli, that you probably know, just to slash the midpiece and get the midpiece, like laser cut the midpiece and then microinject the midpiece. Nevertheless, we didn't have the permission to do that. We have some preliminary evidence in the porcine, and in the porcine, you re-establish a normal spindle if you do that. But that doesn't necessarily mean that you solve the problem. One thing, for sure, you know, this is it.
Well, I'm just wondering, just 1 second, since we're trying to move our activated eggs to a point to get them to cleave. And it does speak of the missing ingredient, which we know from the Amazon and the Senate experiments that were published 2 years ago with human eggs. But they got much more advanced development after they injected pieces of the Senate.
We've done an injection of an isolated tail to inject the donors and what we did, we dissected the sperm and the tail, so we did the injection of the head, and you had some mosaics.
Yeah.
But if you add the tail, it corrects somewhat. Still, it looked like the integrity. Also, the way we isolated it, it was a previous speaker mentioned, and just sonicate, we used sonicate or mechanically. There was some difference there. But that's an interesting concept. We have to find a better way to inject and cut something. But we're sure.
It is technically very demanding, at least in our experience. So,
So, quick question about the techniques. How do you activate the eggs?
yes,
What do you use to activate the eggs? To get the pathology?
I am mycin 5
And
micromolar.
do you suppress the extrusion of the second process?
Sorry,
Do you suppress the extrusion of the volatile?
with 60 men.
Huh?
With 60.
Like a phosphoramide. So when you do that, you really do clone. That's what
Yeah.
they do.
We use 60.
For the whole also.
Most of the cell lines are deployed. Not
Yeah, of course.
Not all.
And the only way they can
Yeah.
proceed. And I think the percentage of blasts to get about the low protein is because some of the imprinting gets
Yes.
screwed up by variability and is similar to a bisexual reproduction.
Yeah.
That's the only one you get to find.
When do they lose chromosome stability? So have you done next gen sequencing or fish to follow these embryos through to see when the chromosome stability is lost?
I don't know, we didn't do this experiments, we just looked at a certain, these were cell lines, so basically we are looking into cell lines, so I don't know, we have this. Excuse me. I know I should stand here, but I cannot hear the question well. So I just try to be, so most of the experiments were done on cell lines. So cell lines, we have some experiments on embryos, and if I remember correctly, the differences are evident from 4 cell stage. Significant, you know, very different number of chromosomes.
Can I ask, so have you had stable cell lines and looked at their stability versus ones that were frozen and then thawed again and then grown? Do you see differences in those? Because there is a thought in the stem cell line industry, basically, that once you freeze them, that there actually is there's a greater instability of cell lines than compared to ones that are just passage continually.
Okay, this is something that happens with embryonic stem cells in general. It's reported, widely reported in literature. For sure, we have differences, not in terms of number of chromosomes per specific line, you know, overall. But we have differences in other features. For instance, sometimes you get colonies that start and boom, they just go. Sometimes it takes forever. So somehow you are dealing with a population that, although very homogeneous, looking at them and in terms of staining and, you know, OCT4 expression and a NOG and blah, blah, blah. In reality, you for sure have subpopulation that have got something which makes them different. But I think that this is taking place in at least all stem cell lines that I have worked with, regardless to the parthenogenetic ones, even the chemically programmed one, IPS. So I think it's really, it has something to do with the cell, the stem cells.
It's because we're not culturing them under any kind of circadian signals.
That's good. That's a possibility.
It's totally possible. That's right. There's no circadian piece to anything we're doing.
We try to keep the condition as steady as possible. Heated stage, constant heated stage all the time.
It's probably on stage.
Oh, well. Okay.
David, quick, is it quick? It's hard.
We actually have babies from mice, the parthenogenetic creation. We published this back in stem cell. By the way, our university was dying to put this out as a recipe. We said, please, no, we don't want this science on it. But we were, you know, this mouse is very robust, strong, and we got babies. We used tetraploid embryo complementation to create kind of a carrier placenta. So that was cheating in the sense because, you know, but we're able to do just as you did with the sheep, you're able to get full form fetuses. They were a little smaller than usual, but full term. And you know, the mouse is much easier to work with. Here's what you have to do.
Can I answer? No.
Yeah, go ahead.
No, I was just saying that the only tissue we found some problem was the muscle. Muscle seemed to be slightly downregulated, but this is something that Fundele at the time also described when he was doing chimeras in the mice. So I think that's probably, that's the part
As
that.
soon as we correct the circadian rhythm.
And yes, and the same.
Okay.
Thank you.
Now, our next speaker in our Italian section is, oh gosh.